Thank you, everybody, especially you, Q, for the invitation, for the chance to come and speak to you today. Um, I'm going to talk to you about work my lab is doing on building a new model of studying biofilms in ventilator tubing. If I can find my slide changer. Yeah, I'm speaking to you today. I'm based at the University of Warwick, which is actually in the city of Coventry in the UK. The reason I'm in the Australian biofilms section is that all of the work I'm going to talk to you about is the result of a collaboration between Warwick and Monash University. So our two universities have a research alliance which has been funding a number of projects related to antimicrobial resistance. And this has led to a lot of collaboration between the institutions. So that's the setting for the work I'm going to present today. And the healthcare context I want to set this in is ventilator-associated pneumonia. So this is something I think that increased in everybody's consciousness with the COVID-19 pandemic. It's one of the most common healthcare-associated infections. And what happens is that if you're unlucky enough to have to be connected to a mechanical ventilator, the way you're connected to that is this very stiff siliconized PVC tube will be put into your trachea. It has an inflatable cuff to hold it in place, and that's what connects you to the ventilator for the airflow. Now, like any indwelling medical device, you've got quite a risk of microbial contamination here. Any bacteria from your mouth, your nose, the environment, once they go into this tube, it's very hard for them to get out. If you're ventilated, you will be sedated, you will be lying almost flat, so you have really no cough, reflex and clearance. And you've probably got underlying health conditions that would predispose you to infection anyway. So having things like chronic obstructive pulmonary disease, diabetes, increase your risk of developing ventilator pneumonia. And once these microbes get in, they form extensive biofilm on the external surface of the tube and above that inflated cuff where any infected secretions will pool. And then with the airflow, they can get aspirated inside the tube as well. So here's just an example electron microscope image of a biofilm on a section of tube that's been taken from a patient. The reason this can lead to pneumonia is that bits of that biofilm fall off and drop into the lungs, and that's what seeds the pneumonia in the lower airways. The incidence of ventilator-associated pneumonia, or VAP, varies quite widely between hospitals, between intensive care units. Overall, if you look at sort of global estimates, you'll find an incidence of sort of 10 to 30% in the global north, but up to perhaps 50% in parts of Asia and Africa. So this is a big healthcare concern. Where I am in the United Kingdom, if you look at the health and economic outcomes of ventilator pneumonia, that will lead to a patient having an average of 6 extra days in hospital, additional care costs of £10,000 to £20,000, and a 30% increase in mortality risk. So this is a serious condition. The problems here is that there's no agreed optimal antibiotic regimen. So prescribing antibiotics for VAP will often be empiric. It won't be based on antimicrobial screening, diagnostics. And the really interesting thing, given that we've just had a talk about wound dressings, is that the development of coatings and materials to make these tubes out of to reduce the chance of biofilm colonization has really not been very successful. And actually that's an understatement. I mean, there's virtually no antimicrobial tube coatings out there. So if you look at the sort of the global market of making these tracheal tubes, these sort of plain PVC silicon coated tubes, it's massive. It's valued at $2 billion US. And given that, and given the scale of that, there's been a huge amount of lab-based research into developing different materials and coatings that could prevent VAP. And you'll see a lot of materials that are familiar from antimicrobial catheters or... Sorry, I can hear some other audio. It's quite difficult to... Thank you. That's easier. Um, yeah, so if you look into the literature, people have investigated different polymers, different metals, for instance, for coating these tubes, but there's been almost no translation into the market or to the clinic. So until the end of last year, there were really two functionalized tubes on the market and in clinical use. Sorry, I can still hear an echo. Is it possible to... Thanks, UQ. Okay. Ah, fantastic. I can no longer hear myself. That's great. So yeah, there were these two sort of big companies who made products that were on the market, and these were the TheraShield tubes, and they had a coating that's positively charged, so kind of inspired by antimicrobial peptides. And there was the Bactiguard tubes, and they were coated with a noble metal alloy. But these two tubes, they're not hugely effective in improving patient outcomes, and they are very, very expensive, especially, as you can imagine, the Bactiguard tube. And that was actually discontinued last year. So we're really, if you contrast this with the myriad functionalized sort of advanced wound dressings to prevent and control contamination, the market for endotracheal tubes is absolutely tiny. And I think one big reason for that is we don't really have any good, tractable, valid models to use in the lab to actually test these materials and find out more about the microbiology of the biofilms. So there are animal models for ventilator-associated pneumonia. Those are ventilated mouse and pig models. Now, that's a really severe intervention, very expensive, very low throughput. We're thinking about reducing the use of animals in research. This is something we want to get rid of, really, if we can. And to try and study these biofilms in the lab, there is no defined microbiological medium that mimics airway surface liquid in ventilated patients. So I came to this area having done a lot of work in cystic fibrosis lung infections, where people have developed all sorts of formulations of synthetic cystic fibrosis mucus. They've taken expectorated samples from patients, they've chemically analysed that, and they've been able to build lab media that mimics that environment. No one's done that for VAP, and one reason for that is it's actually quite difficult to sample airway surface liquid. If you're studying CF, it's easy. People cough up sputum, you can collect millilitres of it. If you look at attempts to sample airway surface liquid, you have to do all sorts of crazy things, like put a tiny bit of filter paper on the end of an endoscope and try and brush bits of liquid off the tracheal surface. So it's quite tricky. And we can't really use healthy human airway surface liquid as a model for what's going on in these patients, because, as I mentioned earlier, they often have underlying conditions that will alter the chemistry of that airway surface liquid. So we need to find out what that sort of physicochemistry of that environment is. Interestingly, if you take a section of endotracheal tube and put it in standard laboratory medium, we found it's actually quite hard to get pathogens to form a really adherent biofilm on it. So our standard lab media are clearly not doing the job. So we need to build a new model of this. We need to start by actually creating some synthetic ventilated airway mucus. Then we're going to investigate how different pathogens form biofilm on these tubes in that new medium. I'm going to show you quite a lot of cryo-electron microscopy today, because that's been one of our key tools to look at these biofilms. So this work has really been led and driven forward by Dr. Dean Walsh, who was until recently my postdoc on this project. He now works for the UK Health Security Agency. And where Dean and I started was thinking about this chemistry of healthy and ventilated airway surface liquid. So in a healthy person, you have quite a thin layer of liquid on the surfaces of your airways. You'll have a liquid periciliary layer, and then you'll have quite a thin mucosal layer, which will have some amount of mucin nutrients in it. We did a very extensive literature review and systematic sort of analysis of the different papers where people had tried to sample the airway surface liquid of ventilated patients and chemically analyse it. And what you generally find is that mucus layer is vastly increased, the periciliary layer is compressed, and you have a big increase in the amount of mucin, amino acids, DNA, and microbial micronutrients in that mucus layer, and also a slight drop in pH. So you can see straight away why microbes like to grow in this. What we were able to do based on this was to create a chemically defined lab medium incorporating these components in what we hope are realistic concentrations. And we can combine that synthetic airway ventilated mucus with endotracheal tubing. So we buy that from a medical supplier, we cut it into small segments, UV sterilize it. We coat it overnight with serum because when a tube is put into a patient, you'll get a sort of conditioning layer on that tube of the patient's epithelium and secretions. And then we can infect with microbes, and we grow that in a 24-well plate in 5% CO2. Here's just a quick photo of Pseudomonas aeruginosa. So after 48 hours growth on a tube section, you can see a lovely green Pseudomonas biofilm. And if you stain that up with crystal violet, you can see we've got biofilm adhering to the surface of that tube. So what have we been using this model for so far? I'm going to show you work really that's focused on 2 bacteria and 1 fungus that we know are implicated in many cases of VAP. That's Pseudomonas aeruginosa, which is coming up in around 10% of cases of VAP. Klebsiella pneumoniae, slightly smaller percentage but very important pathogen, and Candida albicans, which is fairly commonly found in that endotracheal tube biofilm in patients. The questions we've been asking over the last 2.5 years have been, how do these pathogens make biofilm in the tube model? Does this biofilm structure differ when we use our synthetic ventilated airway mucus versus standard rich laboratory medium? Does growth in this environment alter antimicrobial susceptibility? I think talks from other speakers this morning have queued that up very nicely, that growth in host mimicking environments can massively increase antimicrobial tolerance. And can we explore the 3D structure of the biofilm matrix and what it's made of in these biofilms? And ultimately, if we can work out from these questions how good, how useful, how realistic our model is, then we want to be able to use this model so that we can find new ways of preventing and treating tube biofilm. If this model is a good one, then if a researcher or a company has a novel tube coating or a novel antimicrobial adjuvant, then this would be the model that you could test it in and hopefully get more predictive results of how it might work in the clinic. So starting with Pseudomonas aeruginosa, these are cryo-scanning electron micrographs of Pseudomonas aeruginosa biofilm grown on the surface of the tracheal tube, either in standard rich LB or in our chemically defined medium, which I'm going to call SVAM. Now, cryo-SEM is a great tool for looking at biofilms because unlike standard SEM, it doesn't dehydrate your sample, and biofilm matrix is 90% water, so you retain that native structure. If we look at the Pseudomonas biofilm grown in LB versus SVAM, you can see some changes, and really we find that there's more matrix relative to numbers of bacteria when we grow them in the SVAM, so something is changing for Pseudomonas. Dean cross-referenced that with some confocal microscopy as well, so here you can really see in your side view the increase in thickness and sort of surface topology of that biofilm when it's grown in SVAM. And a little bit more microscopy there for you to see. We see even more pronounced differences when we started looking at Klebsiella pneumoniae. So when we grow them on the endotracheal tube in LB, what we can see is individual bacteria tightly attached to the surface of that tube with pili, and they have a smooth surface. Whereas the difference in SFAM is we really see this matrix encased biofilm here. And when we look at the individual cells, we can see that they've switched to a rough surface morphology. And this is the hypermucoviscous phenotype of Klebsiella, where they start to produce more capsular polysaccharide. So we've really changed the growth of those bacteria. Candida was really, really cool. So you queued up nicely for us talking about sort of the yeast to hyphal switch in Candida. And if we look in YPD growing on our tube surface, you see a biofilm made mostly of hyphae. But in the SFAM, they switched to being mainly yeast and pseudohyphae. And this is very interesting because while Candida hyphae are associated with a lot of clinical biofilms, if you look into the literature on urinary tract infections, growth in urine also triggers this kind of phenotypic switch. So there's sort of a precedence there for yeast and pseudohyphal biofilms being clinically important. So that's quite a big change in that morphology. What about increasing complexity? We've been working with some clinical isolates of these species as well, and we've started to do some work on multi-species consortia, because this is a polymicrobial infection like most biofilm infections, right? If it's going to be a good model, we need to be able to grow multi-species biofilms. So this is work started by Dean and continued by my new postdoc, Dr. Jeff Cheng. And what Dean and Jeff focused on is they looked at two different 3-species consortia. They took two sort of early colonizers, pioneer species of that biofilm tube microbiome. So they looked at Candida albicans, which tends to be an early colonizer, and that thick biofilm it forms can be a nice niche for other species, and Streptococcus sanguinis, which tends to colonize from the oral microbiome. And then they added an opportunistic pathogen, either Pseudomonas aeruginosa or Klebsiella pneumoniae. So here you can see monospecies growth of each of these species on the tube, and we're getting a good high biofilm titer. Here's data from the 3-species consortium with Pseudomonas, and you can see that while Candida numbers have dropped a bit, we are getting stable co-culture of those 3 species. And similar story for Klebsiella pneumoniae. I'm not going to give you much more detail of that. We've spent a lot of time optimizing the inoculation and incubation procedures to get a stable 3-species community, and that will be forthcoming in a future paper. What I want to move on to next is how we've tested whether this model could be used for sort of easy high throughput antimicrobial efficacy testing. So what Dean was able to show was that he was able to fairly easily cut large amounts of sections of tubing, grow a mature biofilm on that for 2 days, transfer those pieces of tubing to fresh SFAM containing antibiotics, and basically conduct MBEC tests in this model. So for lab strains of our 3 focal species, you can see they've got a really high tolerance to a chosen clinically relevant antimicrobial. We've just highlighted here where we see a 3-log reduction, and those concentrations are pretty high. If you contrast that with using a Calgary device, the peg lid biofilm using SFAM, so same growth medium but attaching to polystyrene pegs, then you can see for Pseudomonas and for Candida, growth on the tube surface rather than the Calgary device has massively increased antimicrobial tolerance. The yellow arrow is highlighting the MBEC in the Calgary device. We've then started to look into the biology of the biofilm matrix. So Pseudomonas, Klebsiella, Candida, most of these pathogens involved in VAP, they're opportunists. Do they switch how they make their biofilm matrix depending on the environment they find themselves in? What do they make it of when they're in an endotracheal tube environment? And this is work that's been done by Dean and also my PhD student Io Kade, who's just finished his first year. And that's a collaboration with Professor Kim Hardy at Nottingham University. So most of these species will make their biofilm out of a mix of polysaccharide, extracellular DNA and protein, but we wanted to know, does the kind of ratio of those three different macromolecules vary in the matrix depending on how we grow them. And that will give us an insight into the basic biology of these organisms, but it could also tell us if we want to make an adjuvant to reduce or remove biofilm, what matrix component would we need to attack in order to do that, and perhaps prevent biofilm attachment or potentiate the activity of antimicrobials. So what Dean and IO have been doing is they've been growing biofilms on the tracheal tube in standard lab medium or SVAM for 48 hours. Then they treat the mature biofilm with either DNase, protease or glycoside hydrolases at a very high concentration, so at an excess of enzyme. And then they measure the biofilm load with crystal violet. They've also looked at live viable cell counts as well. I'm just going to show you the crystal violet data today. So Pseudomonas aeruginosa seems to sort of have a fairly similar matrix composition in LB and SVAM. The only enzyme that's causing any dispersal of that biofilm is the glycoside hydrolases, which you can see from the crystal violet absorption here, and just a photo here. You can see how dense that biofilm is as well from how dark that crystal violet stain is, and a little lightening with the glycoside hydrolases. For Klebsiella, we see a bit of a qualitative difference. So when grown in LB, proteinase K and glycoside hydrolases both cause a small amount of biofilm dispersal. In SVAM, it's only the proteinase K that has any effect, and that is a more pronounced effect compared with an untreated control. So that's suggesting that in the SVAM, the protein component of the matrix is predominant, and that's what needs to be attacked in order to disperse the biofilm. For Candida albicans, again, we see a bit of a difference. Proteinase K and glycoside hydrolases both cause statistically significant dispersal of the biofilm in YPD, but for glycoside hydrolases, that's really not very biologically significant. It's a very small amount. Whereas in SVAM, proteinase K and glycoside hydrolase really effectively disperse the biofilm, suggesting that they're using more protein and glycoside and polysaccharide in that environment. So what Dean went on to ask was, could matrix degrading enzymes help to clear endotracheal tube biofilm? And there's precedent for this in wound treatment and also in cystic fibrosis lung infection treatment, where inhaled DNase is used. So Dean again grew the biofilms on the endotracheal tube in SVAM for 48 hours. Then he treated with an enzyme chosen from what he'd seen previously and an antibiotic for 24 hours. And then he measured the colony forming units of both cells that had dispersed into the surrounding medium and cells that had remained stuck to the endotracheal tube. And he did this rather than crystal violet, because if you're going to try and treat an endotracheal tube infection, you don't want to just disperse the cells so that live bacteria go into the lungs. You want to disperse them and kill them. So here's data for Klebsiella pneumoniae, which was looking very promising. So without an enzyme, you need 256 micrograms per mil of colistin to cause a 3-log drop in total viable cells, biofilm plus dispersed. But if you add proteinase K, you can take that down to 64 and get almost complete eradication of that population. Candida albicans, here's an example of one that didn't work, where we tried the glycoside hydrolases, and actually they just disperse. They don't actually help killing at all. They just move the bacteria. They don't eradicate them. But we did find some other effective combinations, and that is all detailed in the paper that Dean has published about this model. Then really focusing on the Monash collaboration, we worked with Anna Traven, and particularly with her postdoc, Dr. Claudia Sim, who came over to Warwick for a few months for a visit. And Claudia was really interested to see if gladiolin could potentiate the activity of the antifungal amphotericin B against Candida albicans. So amphotericin B is a really commonly used antifungal drug. It's effective, but it has issues with off-target toxicity because it can affect mammalian cells. So one strategy to sort of improve the therapeutic window of amphotericin B is to find potentiators that would enhance that activity, enable you to use a lower therapeutic dose of amphotericin B. And what Anna's lab, along with Greg Chalice, who has a joint appointment at Monash and Warwick, have been looking at is gladiolin, which is a polyketide antibiotic produced by a Burkholderia species. So Claudia grew her biofilms of Candida albicans on the pieces of tube, and then she treated them either with a mock treatment with gladiolin alone, which you can see causes no dispersal in this example, amphotericin B, and you can see it started to move some of that biofilm, but you haven't got total clearance, and then a combination of the two, and you can see in this example she's starting to see some clearance. And the quantitative data, which is available as an advanced online publication, she's tried a few different doses, and she is finding combinations of those doses where she gets potentiation, she gets synergy. So co-treating with gladiolin and amphotericin B is enhancing the clearance of the Candida from that tube. So what's the next step? I've been talking about trying to make these biofilms more realistic in the way we grow them in the lab, and what I've shown you so far has one big gap from reality. We've been cutting those sections of tube, sitting them in a culture dish, and just leaving them static, whereas in a ventilated patient, they're being ventilated. You're going to have constant flow of air that's going to affect movement of the fluid in the airways, and so that biofilm is going to experience changes in oxygen regime and also different shear forces, and we know that shear forces can affect how microbes form biofilm. So what we want to do is to actually build a dynamic model in the lab. We're working with Neve Engineering, who are a small robotics and 3D printing specialist company, and what we're doing is we're going to build a 3D printed ventilator, which will basically be a bag valve. So you'd have robotic arms controlled by a microprocessor that will squeeze and release a bag that will supply sterile air plus CO2 to this series of biofilm reactors, where we can also set up a drip flow of synthetic airway liquid medium. We made the happy discovery that a 50mm falcon tube is a very similar size to an adult human trachea. So when you inflate that, inflate the balloon on an endotracheal tube, it will sit very nicely and tightly inside a falcon tube. So our bioreactors will be tubes inside falcon tubes with a waste outlet. And again, we're working with Neve Engineering to work out how we can 3D print reusable mounts for these, so that we can set this all up in the lab and have a reusable, sterilizable, bespoke system. So this work is just started, and it's funded by the National Biofilms Innovation Centre. What's our to-do list next? We need to validate this with patient samples. As other speakers have said today, that's crucial. There are no really good published microscopy images of endotracheal tube biofilms, so we're having to prospectively collect our own samples. So hopefully that will start happening over 2025, and we will be able to compare the structure of biofilms in our model with patient samples. We'd really like to start testing some novel tube coatings, so we're going to try some antimicrobial polymers that we've been working with and coat them onto the tubes. What I'd really like to do is to have this model published, open access and available for academic use totally freely, but also work with contract research organisations who might want to make a version of this model that would be usable under ISO for commercial antimicrobial testing. This is something we did quite successfully with a previous ex vivo model of cystic fibrosis lung biofilm, and we did transfer that to a company. So it would be lovely to be able to do this again with this model. And I just want to finish by stressing the 3Rs potential of this model as well. This is something that I think really could replace that reliance on mouse or pig models. So summing up, we made a new chemically defined medium that mimics ventilated airway mucus. We've successfully used it to grow biofilms of key pathogens on sections of endotracheal tube. And we're now moving on to see if we can look at pieces of tube connected to a ventilator in this model. We've shown that our medium cues clear differences in biofilm structure and matrix composition compared with standard rich lab medium. And we've published the composition of that medium, so you can use it. And we've confirmed that VAT pathogens are much more antimicrobial tolerant when they're grown in our model compared with a Calgary device. We very much hope that this model could be used in preclinical testing to help discover new tube coatings, drug candidates, drug combinations. So thank you very much for your attention. Quite a few people contributed to this research, and again I'd really like to stress how much this has been driven by Dean Walsh. His work's being continued by Jeff, and also Matt, Io, Jennifer and Kat through their studies have really contributed to this work. We did consult with clinicians at the University Hospital who've been really great in helping us make sure this is as realistic as possible so far. Cryo-SEM, thank you to Saskia and Chris, and thank you to all of our collaborators at Monash and in various other places. So I will finish there. Thank you.
Thank you so much, Freya. Is there any burning questions? Otherwise, I might actually just wrap up this session just because we are behind in time. Is there any questions? No. Oh, wait.
Just,
yeah.
Thanks for joining us. I actually met Anna on Monday. Just add one comment on the antimicrobial coating. So recently we put out a few infected and uninfected ECMO cannula from the patient. And what we found is either they are infected or uninfected. If you put them in for 72 hours, the whole surface will be coated heavily by the fibronectin or collagen, a layer. It's very clear, it's a layer of stuff on the surface. And we do, on the infected drug under the cannula, we did find biofilm grow on the top, on that coating, on the like fibronectin and the collagen coating, rather than the smooth surface of the device, of the cannula. So it is possible that coating, if you even you have the coating, it will be masked by those and like actually what we call matrix protein, like collagen fibronectin. So it won't work. This is our thoughts, and it's based on our clinical finding. Do you reckon?
Yeah, I think that's a really good point, and we're kind of trying to mimic that coating just using serum, which obviously isn't as complex as the full fibronectin. My gut feeling is that you're absolutely right, and I think where antimicrobial coatings could potentially help is preventing the initial contamination of that tube as it's being put into the patient. And as you say, once you've started to get deposition, then it will become much more difficult. Clinicians do try to clean out that deposition layer in the tube, so they literally put a very fine brush down into the tube and pull out anything that sticks. So I think you'd also have to look at whether that is clearing enough of that host material coating that the actual tube coating would be visible. But yeah, I suspect that is one reason why the coatings are not massively successful.
All right, let's thank Freya again and thank all our speakers from this session and the Australian segment of this entire program. And yeah, we'll thank everyone and have a good afternoon. Bye.